All right, hello. We are doing the last lecture in the first unit here, lecture 8. And this is going to focus on viral replication and damage. So in the last lecture on viruses, we did an introduction. And so here we're going to talk more about the way they replicate, and then that leads to the way they cause damage. Let's start with the replication cycle. This is specific to animal viruses, so that you understand, and there are plant viruses and bacterial viruses, and they're all a little bit different. So this is animal viruses here. There's 6 stages of this replication cycle. You'll want to be familiar with each of them. You'll also want to be able to put them in the correct order. So the first one is called adsorption. It's not a typo. It's not AB, but AD. And it's sort of a fancy way of describing attachment to a surface, and we'll describe that one next. Penetration and uncoating often go together, although they're 2 separate steps. This involves the virus entering the cell and shedding the capsid or any kind of coating like the envelope in some kind of fashion. And so that gets rid of all of the protective layer, getting down to really just the core genetic material in the virus for the most part. Synthesis and then assembly take place where the virus now hijacks the host cell machinery. So this is where, for example, ribosomes are utilized along with the Golgi apparatus, and that's going to allow the virus to sort of synthesize and assemble its own particles using the host cell as the sort of factory of the machinery, which is why it needs to get into the host cell in the first place. So really adsorption, penetration, and encoding are all sort of prerequisites to get you to synthesis and assembly, which is now kind of the reason you're in the cell in the first place. Finally, then release is just a mechanism to get out and spread to the next victim. So there's a couple different ways that works. So like I said, be familiar with all these here. Adsorption is a key aspect that we're going to talk about next. Penetration and encoding are important, but not too complicated. We're going to really skip past a lot of the detail in synthesis and assembly because that could take all semester, but there will be a few points here and there that are worth noting, and then the book will go into a little bit more detail on that as well. And then with release, we see kind of how viruses get out, and so there's some emphasis on that as well. So we're going to not give it as much detail on these steps as what we should or could, but we're going to kind of go through each step in some more little detail or others. So adsorption is the first major step, and this will be a key emphasis here. So this means adsorption rather means to adhere to a surface. In this particular context, what that's referencing is how the virus attaches to the host cell. And the way that it's going to do that we'll discuss next. But what you basically want to understand is that this is a prerequisite to all the other steps. So one of the reasons why it's important to understand what steps occur in what order is that step 1 has to be completed before step 2 can begin. Okay, and that may sound obvious, but just to clarify. So as it says here on the last line, viral infection is not possible without proper adsorption. So this is a key limiting step. And just to add one more thing before we go on, we are inundated by viruses every day in numbers that are shocking. The textbook talks about some of the numbers if you want to look in the first couple of sections there. Trillions of viruses. The vast majority of those viruses cannot adsorb to our cells. So this is a key limiting step. And this is what maintains certain viruses in the animal world, certain viruses in the bacterial world, and certain viruses in more specific sectors. Protozoa, fungi, algae, they all have their own viruses. And adsorption, for the most part, kind of keeps them in their own little sector. And so you don't usually see viruses jump from one kind of host species to the other. Adsorption usually keeps that sort of in check. Not perfect, but that's where this is a key step. Now, the mechanism of adsorption is here. What you have is on the virus what's called a spike. Spike protein, which is specifically a kind of protein called a glycoprotein. Glyco is in reference to a carbohydrate or a sugar, and so this is sort of a carbohydrate sugar protein, and it's a specific kind that's often used in cell surface proteins, identification and things of that nature. So this specific kind of protein then has the capacity to only bind certain interlocking structures. Therefore, the exact shape of the glycoprotein is going to act kind of like a lock. And so that analogy of locking key specificity comes back. We've talked about that earlier. kind of a fundamental concept that happens in different ways all throughout biology. So here we see that mechanism of lock and key interaction where the cell has this glycoprotein, and that should be labeled there better. So just to clarify, this little field goal looking thing, that is a representation of a glycoprotein. That's basically a lock, and we're simplifying it here to see it, but the key then has a counterfeiting shape to fit in there. That is actually the virus spike protein. So those probably could be labeled better, spike and glycoprotein. So that is essentially the mechanism that the virus needs to get into the cell. They got to have the right key to get into the right lock. There's a video that I have in the virus video assignment. It's the one of the short videos it's the second video. It's a short video that shows an animation of a virus attaching to the host cell. I forget what that's called now. How a virus attacks a cell is what that was called. And anyway, that animation is relatively short and it's relatively simple, but it really does a much better job of illustrating kind of what this looks like. And then other videos like the HIV video do that as well. There are a lot of key details that can occur with each virus and exactly how that works. But that's sort of the general idea. And one more thing here, this key, by the way, can mutate. And that's one of the things that you see with questions about different viruses and how they might jump from one species to another, potentially. And where that could theoretically happen is that the key can actually mutate, and the viral genetic material changes. That changes the key over time, and now all of a sudden it fits a different lock better or worse. And the idea is that maybe it fits a new lock that it didn't used to fit, and now the virus can maybe jump to a new host. And those are theoretical and proven ways, actually, both, that viruses can actually potentially become zoonotic, transmitting from one species to another via mechanisms in the viral spike and the changes that correlate with different configurations of the host glycoprotein. Okay, in this section, we're going to make just a really simple diagram, just to really illustrate the point and hopefully overly simplify it. So let's start by, in the space that you have there, drawing a virus. And these are just going to be kind of a simple, arbitrary kind of drawing. So here's our virus, and these represent the shape of the spike proteins on the virus. And I went with just simple shapes here. Viruses would, in all reality, have many more, but just for simplicity here. Okay, now, what I'm going to show you next are drawings of 3 potential host cells. And then what I want you to do is take just 10, 15, 30 seconds and just examine the shapes of these cells and think about which of these cells would be potentially infected by this virus. Okay, so here's host cell A, host cell B, and host cell C. I've attempted to make these drawings somewhat representative of interlocking shapes. So if you look at the receptors, these represent the host glycoprotein receptors. And we have 3 different host cells, each host cell with its own set of receptors. These are representing just a small fraction of the number of receptors that a cell would have. So based upon the shape that you see here, which of these receptors here would you suspect that the virus would use to infect the cell? And if you need to pause it right now, go ahead. But the answer is going to be host cell A. And if you look here at this receptor, this is going to match with the box kind of configuration. So all these other receptors don't really match exactly, as the illustration is attempting to show there. And so based upon this diagram, this virus would be not only able to infect host cell A, but only through this one receptor. Okay, so this sets up what is known as the host range of a virus. So the host range and adsorption, lock and key specificity, these all go hand in hand as a sort of broader concept. The idea is that we're inundated with viruses all the time, but only a small fraction will actually infect us. That's because each virus will only match within the host range. So these are all generalizations. Not every virus is so easily to put in these categories, but there's a few examples, at least one example for each of these here. Restricted host range would be where a virus only infects one or maybe a very small number of cells, usually within one species. This is restricted host range. The example here is hepatitis B that only infects liver cells. Another example not listed here would be HIV that generally only infects CD4 helper T cells, which is mentioned in the video. Moderately restrictive is probably where most viruses fall closer to. Poliovirus is an example here. It can affect both intestinal and nerve cells. And some of these viruses are known to even sort of infect a wider variety of cells throughout the body. Broad host range is relatively common as well. This is where a virus can infect different species and jump from different animals. So you do see this with certain viruses like COVID, which can actually infect a wide variety of different animals. Influenza viruses do this as well. It causes them to mutate at a higher rate as well. But the example listed here is rabies. Rabies is another good example where rabies can actually infect most mammals with a few exceptions, like possums, which usually don't carry it because their body temperature is too low for the virus to replicate successfully. So there are certain restrictions that sometimes go outside of the lock and key mechanism, but the idea there is that a virus will sort of fit into one of these three general categories based on the degree of its host range. So that kind of explains a little bit about why some viruses are more widespread and some viruses are real specific to what they do. Once the virus has successfully adsorbed to whatever cells it can, then the next thing it will attempt to do is to get into the cell. And for animal viruses, this will occur in one of two ways. One of those is called endocytosis. What happens here is that the virus is engulfed and enclosed in a vesicle, as shown here. This is actually a natural process. Cells often use endocytosis and this vesicle formation to bring in things like nutrients. And the virus has tricked the cell into doing it in a way that allows it in rather than some natural substance. Fusion of the fusion mechanism takes place with certain types of viruses called enveloped viruses. We talked about those in the last section. I mentioned naked viruses and envelope viruses. We'll talk a little bit more about the envelope in a second here in the coming section. But what happens with envelope viruses is they have this special layer called the envelope, and it's the same material as the host membrane. So what ends up happening is you have these two almost nearly identical properties of material here, and because they're so similar, when the virus binds, the two actually just blend or fuse together. And as a result, the membrane of the cell and the envelope of the virus become one. What that does then is it slides the capsid, which is beneath the envelope, inside the cell. And then from there, the virus will shed the capsid to get the RNA or DNA, whatever nucleic acids it has. So these are just sort of two ways that the virus gets in, and you just want to be familiar with the general differences between the two. So just to repeat, endocytosis involves the formation of a vesicle. That's kind of the key idea here. And then fusion only occurs with envelope viruses, in which the envelope fuses and becomes part of the cell membrane. Now, uncoding can be described here in relatively simple terms. If you're looking at complex virology, this could be a much longer discussion. But in simple terms, uncoding is just removing the capsid and exposing the central core. And the central core would be any kind of nucleic acid, or if there's any sort of enzyme or protein that the virus may require. All of that is shed. So at that point, then, the virus has successfully entered its host cell. It's adsorbed, which is the most difficult part. It is penetrated, which can be tricky. And now it's got all the core components into the cell. And that is essentially the most difficult part of a virus's job, and now complete. From this point, the virus will, depending on the virus and depending on the exact scenario, the virus will effectively take over the host cell. So a host cell follows the central dogma, taking its own DNA and converting its DNA into RNA and proteins. And what the virus will now do is simply restrict those processes in which the cell is doing that and force the cell to make viral RNA, viral proteins, and then in some cases even copy its own viral DNA, depending on what kind of virus it is. So a DNA virus will force the cell to make copies of DNA. An RNA virus will force the cell to make copies of RNA. And in both scenarios, they'll use the host ribosomes to make viral proteins. And in some scenarios, the virus can live or function alongside the host cell. But in most cases, when the virus becomes active, the normal functioning of the host cell seems to stop functioning. So it seems to be sort of one or the other, although that probably isn't entirely accurate. But synthesis is the process by which the genetic material is replicated and the ribosomes are used to make proteins, to simplify it down. Virus takes what the host is normally doing, making its own proteins, and making its own DNA, and forces the cell to do it for the virus. The virus will then lay down lots and lots of viral particles all throughout the cell. Those viral particles then become assembled into mature viral pieces, sometimes called a virion, which is basically a mature, fully formed particle ready for release. Assembly is another complex thing. There's a lot of different ways that this happens, but to simplify it down, synthesis involves replication and protein synthesis, and assembly involves packaging all those things together into a complete virus. You can kind of see that here where the DNA and then the capsid and then the envelope all have to kind of come together before the last step, which is release. Release is dependent a little bit on the kind of virus that you have. A lot of animal viruses, but not all, but a large number of them will release through exocytosis. This is essentially the opposite of endocytosis. So endocytosis was one way that viruses come in, and exocytosis is one way that a lot of viruses go out. So with exocytosis, you see that with envelope viruses, they line up along the membrane inside the host cell and then push themselves out. And before they fully release, they've also incorporated host proteins virus proteins into the membrane. This is actually shown really well in the HIV video that I'm having you watch. So if you haven't watched that, just make a note that it shows in the HIV virus that laying down these viral proteins in the membrane prior to exiting. So that's a good visualization of what that looks like. So this is part of actually the assembly and the synthesis stage. But those are laid down in advance, and then once the virus is assembled in the capsid form, the capsid pushes up against all of these spikes proteins or proteins in the host cell, and the entire structure merges out of the host cell as a fully formed virion particle. Now, this involves then stealing some of the host membrane in the process. Okay, so this is, just let me go back here, sorry. Exocytosis, as it said, only occurs in envelope viruses. Okay, it involves pinching of the envelope, or of the membrane, excuse me. Therefore, when the envelope virus is fully released, it's actually partially assembled along with the host membrane from the cell that it just left from. So it ends up sort of coating itself in parts of its host, and that is actually what becomes the envelope. The envelope will be a combination of the viral proteins that were synthesized and assembled, but it will also be a combination of some of the very membrane that the host cell itself was made up out of. So that's actually where it comes from, and that's kind of where the idea of an envelope virus is different from a naked virus. So they coat themselves with a portion of the cell membrane. Now, if you're a naked virus or a complex virus, then instead of using exocytosis, there's a different mechanism that can be generally referred to as lysis. There are different ways in which this happens, different triggers, but lysis is basically a rupturing or bursting, and this occurs when the viral particles are mature. Sometimes it's a matter of filling up the cell to the point that it explodes, like putting too much air in a balloon, and sometimes it has more to do with a protein that the virus releases that causes the cell to rupture open. But one way or the other, the cell ruptures, and this is actually a pretty clear mechanism that gets into a minute when it comes to damage, which we're going to talk about next, actually. So that finishes the cycle there with release, depending on what kind of virus you're talking about. So as you can see there with lysis, getting into damage, whenever a cell is involved in lysis, it's relatively straightforward. It kills the cell in the process of release, and any viability that cell had is now gone. If you have lots of viruses infecting lots of cells, they're going to do lots of damage to all those cells within that tissue. If the virus is restricted to only certain kind of cells, kind of like the hepatitis virus is restricted to your liver, then that damage is going to be restricted to the liver. If the virus can spread and move all throughout the body, then that could be relegated to different areas of the body. But you can see here under this illustration, normal healthy tissue here, and then virally infected tissue over here, just a large clearing where those cells used to be. So when it comes to lysis, I feel like the damage is relatively straightforward. You're rupturing the cell, causing damage of any cell involved. Now, just to clarify, your body can recover and regenerate new cells, and this is where it gets a little more tricky. The rate of damage and the health and the viability and the vitality, really, of the host are key factors. If you are young and healthy, you can replicate those cells pretty quickly. If you're older and not as vital, you don't have as much vitality, then the virus can easily outpace your ability to recover. And so that's where those things get a little more tricky in looking at the scope of damage across the population. You're going to see a wide range in differences because of the host immune response to the virus. Exocytosis is a little trickier, but recall this is where the virus pinches out and uses the cell as its own envelope. Recognize here that as the cell membrane is being utilized to create the envelope, that depletes and deteriorates the envelope of the host. So that actually ends up damaging the membrane, albeit a little bit more subtly than lysis. But what you end up with is normal proteins are often removed from the membrane, and then viral proteins are incorporated, and then the host membrane itself is pinched off. And so usually during an infection like this entire cell will be destroyed through exocytosis, even though it's not necessarily in the same way as lysis. These are this up here is an illustration. This over here in the middle, this is actually a real electron microscope image with a sort of stop frame picture. You can see the virus emerging in all the way out in each of those stop frames. These are artificially colored, but other than that, it's real. And this is also a real image over here. This is showing the surface of an infected cell. And you can see some of the viruses emerging and budding out or using exocytosis to come out. And we don't have a good comparison here, but this membrane should be nice and smooth and relatively even, and you can see it's disturbed. And these all down here are viral particles. So this whole cell is being essentially destroyed in a more subtle way, but similar to the way that lysis affects it. Here what you can see are two cells side by side, kind of a normal eukaryotic cell with the nucleus intact here. And then on this side, we can see a eukaryotic cell with the nucleus being destroyed here during some of the assembly of the viral particles. So the nucleus will normally need to be intact for the cell to function. So this cell is now essentially falling apart in real time as the virus disrupts the nucleus. These are all proteins of the virus being laid down. All the normal functioning is now gone, and you're basically converting your cells into a viral factory. All the normal functioning is now gone. This can be more subtle. This is a more extreme image of what can happen, but in theory this can be more subtle, and there's a lot more research on viruses that is not clear. It does actually appear that there are viruses in your body that maybe live alongside your cells and don't cause so much obvious damage. But the way that we normally think about viruses is they cause damage. That's when they get our attention. So those are the ones that we focus on. And we can see with images like this, at the cell level, these viruses are just destroying the organelles of the cell. So this can lead to what are called cytopathic effects, or CPEs. It's more of a technical descriptor of a term, but cytopathic effects are basically microscopically visible viral damage. So it's damage that you can see with the microscope. Do we normally evaluate people that way? Not necessarily. You could do tissue biopsies, but most likely you won't diagnose a virus this way. It's more of a way to study what they do, not so much about diagnosing them, but theoretically you could sort of look at someone's tissue and tell if they'd been infected by a virus if you knew what you were looking at. One thing that happens that are called inclusion bodies. And these are sort of big masses. Usually these are the organelles being destroyed. And although this picture doesn't fully cover it, normally you would not see the nucleus. In this picture you can, but from different images, the nucleus may not be quite so obvious until everything becomes distorted. And this big sort of distortion here can kind of show up as what you would call an inclusion body, usually with a lower resolution microscope. So it looks a little differently at this angle, but that's kind of what you're seeing there is the cellular organelle damage. Those are called inclusion bodies. Syncytia is a specific way that multiple cells fuse together. So going back to this figure here, as the membrane starts to break down, if there's another cell right next to it, which we don't see here, the two cells will sort of fuse together as they fall apart, and this is what you see here, kind of the fusion of multiple cells into one sort of conglomeration there. You can see that a little bit better here in this model of this image of rat tissue. This is rat or mouse lung tissue. So you can see the normal tissue over here, and then you can see some of the clustered multiple nuclei sort of all joined together in this infection here. So this is actually what is the nature of syncytia viruses, like the RSV, respiratory syncytia virus. This is something that young kids and elderly people can get. And so what ends up happening is it causes damage to your lungs, collapsing the alveoli. This white space here is the alveoli where you exchange oxygen. So as that happens, you lose the amount of area to exchange oxygen, and it makes it harder to breathe. So damage can occur for a variety of reasons, but those are general ideas: inclusion bodies, syncytia, lysis, deterioration of the membrane. Now, another sort of couple more general ideas. The body, whenever it spots a virus, turns on the immune system, and this creates inflammation, and our own bodies can even then attack our cells in what's called cellular apoptosis. So the general immune response ultimately causes inflammation, and this essentially makes us feel sick and sore, to put it simply. A lot of the symptoms of a viral infection are actually our own body's response to the virus. That's actually the way it works. Illness, fever, soreness, headaches, the rash, those types of things are all actually the response of inflammation, which is your body doing that to you in response to the virus. A little more complicated than that, but that is actually the case. Now, cellular apoptosis is where then your immune cells will target the virally infected cells and actually kill those cells that are virally infected. And what that does is it makes it so that the virus can't continue to replicate in those cells. So the body basically sacrifices potentially healthy tissue. I guess theoretically they're infected, but it will potentially even compromise healthy tissue if it's not properly controlled. And this is where it gets more complicated. The body wants to come in and destroy infected cells, and sometimes can go too far and can actually destroy healthy tissue or not necessarily maintain that in the right area of the body. So cellular apoptosis is the way your body kind of clears a virus out, and that can in and of itself cause damage. Now, autoimmunity is not necessarily something that is quite as straightforward. This is more of just a generalization that in theory a virus could cause the body to react to the tissue even after the virus has been theoretically removed. This is something that's just potentially the case, especially with latent viruses or long-term viruses in which they could cause flare-ups and things like that. Autoimmunity is much more complicated though, but essentially where the body ends up attacking itself later, even if the virus may not necessarily be a threat. Viruses can go into persistent infections where they don't necessarily always go through the synthesis and assembly and release upon entry into the cell. And this is another area where it becomes a lot more complex, but the general sort of take on this is that a persistent infection will cause some kind of lower level of replication of the virus, and so rather than it spreading and releasing really quickly, it tends to be more delayed in the spreading and a little slower on the synthesis and the assembly. And so you end up with more of a persistent infection that in which rather than a virus that goes through it quickly. You know, a lot of the flu viruses and the COVID virus, they tend to come on quickly and, you know, relatively speaking, and go out relatively quickly. Other types of viruses, some of these listed here, herpes viruses are notorious for this, hepatitis viruses, especially B and C, HIV. These are viruses that are not known to just kind of come in and out of the body relatively quickly, within days through a week. They can actually be persistent for your entire life, depending on the exact scenario. And so these establish what are called persistent infections. The mechanism here, this is actually shown in the video on HIV in reference to HIV specifically, but it's somewhat similar in the other viruses as well. But it shows the function of the enzyme integrase. So pay attention to integrase in the HIV video, and it actually will show you how the provirus is formed, in which the DNA of the virus is incorporated into the host cell, and that can become permanent and will become permanent if the virus is successful. Viruses, in the same way that they have the potential to integrate their DNA into the host cell, they also have the same potential to alter that host DNA. Now, there's another article I have you read this week about called the lurker, viruses in the genome. And this is sort of similar, although not exactly. The idea is that viruses can alter the host cell DNA. And there's different ways that they can do this. The HIV virus integrating, and viruses like herpes integrating into the DNA is one of the more direct ways that they do this. But they can also mutate the DNA through damage and things like that, at least potentially. And so the idea is that viruses can actually induce changes in DNA. And when you look at what is the cause of most cancers, cancer is also caused by a change in the DNA. It ends up being much more complicated than that. To simplify it down, a lot of cancers at least result from a change in the DNA. And so anything in theory that can alter your DNA could cause cancer. And that's why you see a lot of warning labels on things, a lot of chemicals that theoretically can and sometimes are more likely to cause cancer, because they're a chemical that can alter your DNA in various ways. So anything that can alter the DNA can theoretically cause cancer. Some things are very minimal, some things are extreme. Viruses are one of those things that can alter your DNA. And so in the same way that like carcinogenic chemicals can alter the DNA and cause cancer, viruses can alter the DNA and cause cancer. Not exactly the same mechanism, but the same general outcome. So these are said to be what are called oncogenic viruses, and the effects they have are called transformations. Transformations can be highly variable, but some generalities here. Increase in growth rate, changes to the chromosomes, changes to the cell surface molecules like the proteins, and the change in the ability to divide. Actually, cancer cells are known to divide indefinitely, whereas most cells have a cap on the number of cell divisions that they can have. So there's a lot more to be said about that, but viruses are sort of generally thought to be able to do that. And there's a lot of variation in what people think about this, but there's speculation that maybe more cancers than what we currently think are capable of causing, more cancers than we currently recognize are actually caused by viruses. So some examples here, viruses, these are called oncoviruses. The papillomavirus, such as the HPV, human papillomavirus, the herpes viruses, and even the hepatitis viruses, such as hepatitis B and C. So these are all those that can alter the DNA. They can often do so by trying to integrate, changing the code. The code being changed leads to a cancer, which I said is not really something that we're explaining fully here. That's really a whole nother lesson. But cancer can be sort of, the common denominator in most cancers is a change to the DNA for various reasons. And so these are viruses that can all do that. Last line, as it says here, there is a lot of speculation that this could be much more widespread than what we currently are recognizing. And the last thing here is treating viruses, also to simplify this down. Viruses ultimately have to be cleared by the immune system. This is one of the reasons why viruses tend to be more capable of wreaking havoc on people with a damaged immune system. Ultimately, you'll have cells that remove the virus from the infected tissue. These would be your T cells and white blood cells. And then you'll have antibodies that will block the virus from spreading and infecting new cells. And it's usually the combination of your white blood cells and your antibodies that allow you to fully get rid of the virus to the extent that you can. One of the things that you can do, though, in certain situations is introduce antiviral drugs. Antiviral drugs are drugs that in general try to block one or more stages of the replication cycle. So you might try to block adsorption, or you might try to block the uncoating or the penetration or even the release of a virus or any aspect of synthesis or assembly. And so these drugs are all designed to kind of almost like throwing a wrench in the gear of the virus. You're trying to basically slow it down or stop it in the process of replicating. So those can be effective. They are using those for HIV because otherwise HIV or even a healthy immune response can't stop HIV. So certain drugs, it's really key to stop that replication. But for the most part, in most people with most viruses, it really comes down to the immune system to actually be able to clear it. Antivirals are not going to cure a virus. Even with HIV, they've not been able to cure HIV. So antivirals simply stop it from replicating. And that's really good because you can really cut down, and we may actually theoretically be able to eliminate HIV because it no longer is able to replicate. But it doesn't actually remove it from the individual who's infected. It simply keeps it dormant. So there really is no cure other than your immune system clearing the virus. Some viruses are almost impossible, like HIV, but most of the time our immune system can do that. So, all right, I'm going to stop it there, and this will be the end of Unit 1.